Hello everyone, this is GATE 2023 Life Sciences, Section P Biochemistry, question number 39. A linear DNA fragment of 5 kilobase kb when completely digested with EcoR1 produces 2.5 kb, 1.5 kb and 1 kb fragments. Complete digestion of the same 5 kb fragment with XBA1 produces 3.5 kb and 1.5 kb fragments. Which one of the following set of fragments will be obtained if a 5 kb fragment is fully digested with EcoR1 and XBA1 simultaneously? And the options are 3 kb and 2 kb, B 2 kb and 1 kb, option C 2 kb, 1.5 kb, 1 kb and 0.5 kb, and option D are 2.5 kb, 1.5 kb, 0.75 kb and 0.25 kb. This is the NPTEL online course reference in which they have explained the restriction enzyme digestion and how the restriction enzymes are used for rDNA technology. So restriction enzymes are also known as molecular scissors. Restriction enzymes or molecular scissors. So these enzymes are naturally found in bacteria, right? They are naturally found in bacteria and they play a protective role in bacteria. How? So as we know that there are phages which have a double-stranded DNA as a genetic material, and these phages infect bacteria, right? These phages infect bacteria. They utilize the host molecular machinery and nutrition, and they divide themselves, and finally they kill bacteria, kill the host and propagate themselves, right? So in order to protect themselves from these phages, the bacteria also has few restriction enzymes within themselves. And what does restriction enzymes do? They cleave the double-stranded DNA, right? They cleave the double-stranded DNA of these phages. Thus, they kill the phage DNA so that the phages no longer will infect bacteria and utilize the host molecular machinery. Okay, so this is the role of restriction enzyme inside a bacteria. So, in rDNA technology, in recombinant DNA technology, we use this molecular scissors or restriction enzymes to cleave DNA of our interest, right? To cleave DNA of our interest for various applications like cloning, any gene, expression of proteins, etc. Okay. So, how this restriction enzymes recognize the DNA sequence? So this restriction enzymes has a sequence specificity. They will recognize the particular sequence. It could be a palindromic sequence, and they will cleave either like this. This is a sticky end cleaving pattern, or they'll cleave a double-stranded DNA creating two blunt ends. So either the restriction enzyme will give us two sticky ends or two blunt ends. It all depends upon restriction enzyme. Okay. So coming back to the question, in the question they have mentioned there are two restriction enzymes, EcoR1 and XBA1, right? So these two restriction enzymes are cleaving a linear DNA fragment. Okay, this is very important. The DNA fragment here is a linear DNA fragment. Suppose this is our linear DNA fragments of 5 kb. So when eco R1 is used at a time, it is giving us three fragments, 2.5, 1.5 and 1 kb, right? So it is giving us three fragments, 1, 2 and 3. For example, let's consider this, and these fragments are 2.5, 1.5 and 1, approximately, right? Approximately these are the sizes. So as you can see, when it's a linear DNA fragments, and if we are getting 3 fragments, we should note that there are 2 recognition sites of eco R1, right? So eco R1 has 2 recognition sites on this linear DNA fragment, and hence we are getting 3 fragments of 2.5, 1.5 and 1. Whereas XBA1 we are getting, when it is digested with XBA1, we are getting 3.5 and 1.5 kb fragments. So we are getting only 2 fragments, 1 and 2, which are 3.5 kb and 1.5 kb. Total is 5 kb, as it's the same fragment. So here we have only 1 recognition site of XBA1, whereas eco R1 has 2 recognition sites on this linear DNA fragment. Okay, so if we had a circular DNA and we got 3 fragments of the same circular linear DNA, then there would be 3 recognition sites of eco R1. Otherwise we won't get 3 fragments. So in the linear DNA there are 2 recognition sites and we are getting 3 fragments, right? This is simple. So in circular DNA there would be 3 recognition sites to get 3 fragments. Similarly, XBA1 will have 2 recognition sites to get 2 fragments. Yeah, this is clear. Also, it is very important that you understand that in the question they are saying it is producing 2.5, 1.5 and 1 kb fragments. So it is not necessarily that they are producing fragments in specific sequence. So in the question they are mentioning it 2.5, 1.5 and 1 kb fragments. So we shouldn't assume that these are the specific sequence in which the fragmentation is done on the linear DNA fragment. Okay, because there is no direction given to us. Direction of the linear DNA is not given to us, right? Also they haven't mentioned any specific sequence recognition that it will be cleaving in the far right end or it is cleaving in the far left end. So this sequence specificity is not given to us. Also they haven't mentioned the fragment sequence, right? What I mean by that, so specific sequence of the linear DNA and direction is not mentioned. That I mean the 5', 3' direction is not mentioned, and they also haven't mentioned the fragment sequence. That is, if from 5' end we have 2.5, 1.5 kb or 1 kb fragment sequence, this fragment sequence they haven't mentioned. So these fragments could shuffle inside this 5 kb DNA fragment. What I mean by that is we could have a linear DNA fragment with 1 kb here, 2.5 kb in between and 1.5 kb in between. So we cannot assume that this is the sequence of the fragments which have been cleaved. Similarly, same goes for XBA1 enzyme. Okay, so there are only 2 fragments. So there are 2 possible combinations. two possible fragments, two possible combinations of fragments could be obtained, right, in response to eco R1. And in eco R1 we could have, there are three different fragments, so we can have three different possible combinations with each other, right? So it is very important that we need to arrange this cleaved fragments in specific order so that we could get one of the correct answer from these options, right? So let's start. So I have so for the reference we'll have the options here, and we'll start digesting the DNA fragments. Okay, so if this is a 5 kb DNA, so first let's go with eco R1. So we have a 2.5 kb fragment, 1.5 kb fragment, and 1 kb fragment, right? And the XBA1 restriction enzyme is giving us one 3.5 kb fragment. So if this is 2.5, this will be 3.5 kb and 1.5 kb. So if we cleave these two enzymes simultaneously, from here we'll get, if this is our first case, we'll get what, 2.5 kb, we'll get this fragment which is uncleaved properly. Then we have, so the 1.5, so we'll have 1 kb fragment from here, right? So this is not a fragment, so we'll have a 2.5 kb, 1 kb fragment, right? And we'll have a 0.5, we'll have a 0.5, and finally we'll have a 1 kb fragment here at the end, right? So this is not matching with any of the options, so this is not the case. So now we have to shuffle these fragments, right? So let's shuffle the eco R1. So 2.5 will bring the 1 kb fragment in between, and 1.5, right? We can do this shuffling at a time. You can shuffle two fragments or the upper XBA1 fragments also. It's depending, like, so if you don't shuffle the XBA1 fragments, then it will have a 3.5 here and 1.5. So from here we'll get 1.5 bands. Okay, we'll get 1.5 band. So and then we'll have 1, this 3.5 is divided into 1 and 2.5. so as you can see, this is also not in our options. Now let's shuffle this one more time. If we take 1 kb here, if we take 2.5 kb in between and 1.5 kb, and let's start with 1.5 kb of XBA1 fragment and 3.5 kb. So here we'll have this 1 kb fragment right from here, and this is whole 1.5 kb, and we have 1 kb fragment, so we'll remain with 0.5 kb here. This is 0.5, and 3.5 is divided into 3.5 is divided into 2.5 and 1.5. So we'll have a 2 kb fragment, right? From here we'll have a 2 kb fragment, because a 0.5 is fragmented from here, so this 2.5 will become 2 and we'll have 1.5. So this is matching with option number C. So in C we have 2 kb, 1.5 kb, 1 kb and 0.5 kb, right? So the correct option is option C. Okay, so if this is a 5 kb DNA fragment, this is 5 kb DNA fragment, right? We have one restriction eco R1 site here and one restriction site here. These are the restriction sites of eco R1 enzyme, whereas XBA1 is cutting here, and we have, this is XBA1 and 3.5. so this will give us these fragments. Okay, 2 kb fragment, this is a 2 kb fragment, 1.5 kb fragment is this last, 1 kb fragment is this first, and this is 0.5 kb fragment. Okay, so the correct option is option C. Thank you.